So what I thought I'd do today is just take a few minutes to kind of set the stage for what I expect and fully expect will be a highly successful workshop. So let's go on to the next slide. I should just say that I've been talking about, as Michelle indicated, I've been talking about exposomics for quite some time. And, oh, here we are. Great, I can actually do this too. So I've been talking about exposomics for quite some time with my colleagues at NIHS, with my colleagues across the NIH. In fact, I had my first one-on-one with Dr. Bhattacharya soon after he became the director of the NIH. And I talked about my personal passion about, if we really want to influence human health, we have to integrate the environment in the exposomics framework into the way that we study the etiology of human disease. So I told him that was my personal passion, and I was very pleased that I think he was very supportive of that. So we'll have to see. So in this meeting, we can actually put together the details and then hopefully come out of this with that framework of what does the Human Exposome Project look like. So let's start off with, I just thought I would, again, to set the stage for a productive workshop, let's talk about some of the grand challenges in medicine and biomedical science. And these were things that I actually took from the back of my brain. I grew up as a molecular biologist, a geneticist, and so these are some of the things that were percolating through the community back in the early 1980s when the Human Genome Project was launched. We really have to get, first of all, we have to get beyond, beyond doing statistical averages as a way of assessing human health. So let's address individual biological variability. I mean, we know in the environmental health sciences community that different people respond to the environment in different ways. So we need to develop a strategic framework that incorporates individual biological variability across the population of humans. And the second and third are really about, can we develop the molecular biomarkers that can complement the ability to both diagnose as well as treat human diseases, as well as to promote human health, assess human health, and promote human health. And the last point here is really important. What about those prevention strategies? If we really want to impact human health, let's come up with strategies based on, individual strategies based on an individual's unique biology that prevent disease from happening in the first place. So with that in mind, I remember as a graduate student in the early 1980s, the community coalesced, similar to what we're doing today, coalesced around this concept of sequencing the human genome. And in February of 2001, there were a couple of very remarkable papers that were published, one by Venter and his colleagues in Science, and the other by Eric Lander and his colleagues at Nature. And this was the human genome. And I don't think anyone will argue that the access to the human genome sequence and the tools that emerged as part of the Human Genome Project have truly revolutionized genetics research. Now, when we look back at the Human Genome Project, it really started, and I think there's some elements here that may apply to developing the kind of the framework for the Human Exposome Project. So the Genome Project, as I remember it, it really started with a grand vision. So if we have these challenges in health and medicine and medical research, what is it we can do? What's the one thing we can do that would absolutely be transformative? So what emerged with this idea that if we go in and we sequence the human genome, that would truly provide us with those transformative set of tools, data and tools. Now, as part of this project, starting with that grand vision, everyone can look at the vision and say, I can get around that, and I want to do what I can to help to make that happen. So it's also, there was the time frame. The Human Genome Project had a beginning and an end. So when does it start? When does it end? And most importantly, I think there was a recognition that we need some money to do this. Grand vision, sequencing the genome, we need some money. And that was the basis. I think it was $2 billion at the time. We can go to Congress then with a very well-defined budget number and good reasoning why we need this money. Now, it's also, I think it became clear at the beginning, that we need to advance the development of new mapping, sequencing, and informatics tools. I remember as a graduate student, there's sequencing DNA with maximum Gilbert, saline-deoxy, these thin gels, and I'm thinking to myself, they're going to do what? And they're going to do in what time frame? Clearly, someone's going to have to come up with innovative, dramatically different technologies to actually get the job done. And I think that's actually what happened, starting with that grand vision. It's what happened in the genetics and genomics community. Now, we also started, the genetics and genomics community, we started with the existing tools. In fact, I think the idea was, let's get beyond one gene at a time. So much of what, in the early 1980s, we'll study a gene. I was a graduate student, I was studying growth hormone, sequence growth hormone. But if you really want to understand the implications of human biology, genetics and human biology, most things are not single gene traits. We have to look across the genome, get beyond one gene at a time. And the last point here was really important. Once the genetics and genomics community decided that we can't compete with each other, the United States can't be competing with the European Union, with Japan and other countries. Once we pull this together, we start working in unison, and we start collaborating, then the job got done. So I think there's some elements here that we can think about, and clearly you heard a lot of those in some of the other introductory comments already. So let's think about, how do we work together? What do different countries bring to the table? And then can we develop this bold vision and then a strategy, operationalize exposomics in a way that it truly becomes a tool that we can integrate into studying the etiology of human disease. Now, I think when we look at just some of the successes, the things we can do today with genetics and genomics are truly remarkable. I mean, the example that I give here is the whole GWAS. We couldn't do GWAS in the early 1980s when I was sequencing growth hormone. But now we can actually come up with polygenic risk scores. So we can actually look at these numbers, and we can predict then how susceptible an individual is to type 2 diabetes based on the allelic variants. Now, it's not just one gene, it's multiple different genes, and the Genome Project gave us the tools to do that. So we can do certain types of things, and of course, looking at the entire genome, we can start an experiment now with knowledge of the entire genome sequence of an individual. So it's truly remarkable. But if you look at, one of the problems is that we can generate polygenic risk scores, but when you actually then try to associate that with predictions on whether someone will actually develop type 2 diabetes, it falls short. It's not 100% predictive. And part of the reason for that is, as a genetics and genomics person, we were studying on the right-hand column here, many of those behavioral problems, ADHD, cancers, immunotoxicity, neurologic function, Parkinson's disease. We were actually studying all those from a genetic perspective. What are the genes and the sequence variations of the genes that predispose to these phenotypes? Now, what many of you in the environmental health sciences community recognized is that there were associations with different environmental exposures: lead, arsenic, PFAS and immunotoxicity, flame retardants, pesticides, PM 2.5. In fact, if you look just at air pollution and health, there are respiratory, cardiovascular, neurological, immune, metabolic, and neonatal effects. So clearly, just by sequencing the genome and identifying those polygenic risk scores, it's not going to get us over the finish line, because there are environmental contributors that will ultimately determine whether or not someone develops type 2 diabetes or other health conditions. So the future is really in working collaboratively together. As Roel very nicely said, the geneticists have to now step in and work together with the environmental health sciences and vice versa. So we need a model going forward where we work collaboratively together. So when you look at the role of the environment, I think we all recognize that we have to get beyond one gene at a time. Okay, I think that was really what Chris Wilde was promoting back in 2005. If we want to understand the environment, there are all of these different physical, chemical exposures. And we're not exposed to just one of them at a time. So many of us sitting in the audience are breathing in those flame retardants in the fabric in our chairs. We probably were breathing in some of that PM 2.5 behind the DC bus on the way here. So we're exposed to all of these things. And it's not just the physical chemical environment that we're exposed to. It's also, we have to factor in, we need a framework to factor in the social, the lifestyle, the ecosystem variables. Physical activity, sleep behavior. Sleep is a wonderful thing. You go to sleep, you're tired, you wake up, you're feeling a lot better. Diet. I know that our Secretary Kennedy is a big focus on diet. It's not just bad diet, but it's also what can we be doing in terms of good diet? And are there things in lifestyle, good physical activity, good diet, lack of smoking that can actually mitigate some of the negative effects associated with physical chemical exposures. So we need this framework where we toss in all of these different variables. So it's in many ways, the Genome Project was simple. All you have to do is sequence the DNA. But here we have all these different variables. We're going to have to think creatively of how do we develop this framework to ultimately study this complex interplay of different environmental exposures. So we need to start, for the Human Exposome Project, let's start as the Human Genome Project did. What's the bold vision? Let's come up with a bold vision that we can all look at, that our legislatures and different policy non-scientists look at that and say, wow, that is really cool. And it's clearly something that's needed, and I want to get behind that. So we also need a definition of the exposome. Now, you got a little peek of that, and I'll take this up in the next slide from the Banbury Conference. We also have to build technologies, the technological capabilities. So once we have the framework, I think it's pretty clear that we can do certain things now. In fact, we talk about, many of you talk about the pragmatic exposome. Let's start with some of the things we can do now, just like the genomics community, getting beyond one gene at a time. Some of those cDNA, spotted cDNA microarrays on microscope slides, it was very primitive by today's standards, but at least it allowed the community to start moving forward. And most importantly, it's the last point here, is we need to establish an unprecedented level of international cooperation. So as Michelle indicated, my passion is collaboration. Innovation, think clearly, differently, to take on bold challenges, and then let's work collectively. Let's join forces, and we can bring our complementary capabilities to bear on solving complex problems. Now, NIEHS, working together with several other ICs that you can see in the lower right-hand corner, and I think this has expanded now way beyond just the institutes and centers on the slide, but it's about coming together and providing some money. And so we decided that we wanted to have one center, and that one center funded at Columbia, Harvard, and the University of Southern California. Their job is to coalesce the global community around the concept of exposomics, build the global community of practice for cooperation and collaboration and coordination with the European Human Exposome Network, develop the conceptual framework to address exposomics, promote technologies and methods and data sharing tools. So capitalizing the existing exposome research initiatives, infrastructures, and resources. Now, we've already heard about this. The first step was let's define what is the exposome. I think that's a very important step. So that was done, actually, with Gary and Michelle's leadership, pulled many of you together. It was a very productive meeting, and what emerged from that was some definitions. We'll be hearing a lot more about that over the course of this meeting. But I think it's clear, exposomics, the common definition, a transdisciplinary field aimed at a discovery-based understanding of how the exposome influences biology and health. So just in closing here, I want to emphasize the point that the genetics and genomics folks have made tremendous progress. So we can now do those GWAS experiments. We can look at entire genome sequences. It doesn't get us over the finish line. And the exposome, once we have an exposome framework, we can look at the influence of these environmental exposures, not one at a time, but collectively, and to do this over the life course. But where we really want to be is at the intersection of both, because it's really, we need a research framework for genes by the environment. And I'm very pleased to see, I know Josh Denny, who's the Chief Executive Officer for our All of Us program. It's a million person, they're not quite to the million, but they're getting very close to that. The idea is to go through and sequence the genomes, collect all of the genetics and genomics data, but both Josh and Jeff Ginsburg, the Chief Scientific Officer, Medical Officer of the All of Us program, recognizes that if they want to bring that program over the finish line, they have to integrate the environment into the fabric of what they do. So they've got the genomics, they've got wearables, physical measurements, but you'll see that the piece of the pie, the environmental data, they're very interested in working with us. And once we operationalize the exposome, we have a willing partner waiting immediately to integrate the operational details of the exposome. So let me just close by referencing the science paper, which hopefully all of you have seen. If you haven't seen it, then I suggest that you take a look at, 2 or 3 weeks ago it was published. These are the Banbury Exposomics Consortium. And I'll quote I think it was the last sentence, a couple of sentences in the article. It's life exists at the interface of genetically encoded processes and environmentally driven realities. So too should the biomedical enterprise that studies it. And this is this wonderful diagram from their paper. So I wish you all, actually all of us, we can take off our coats, loosen our ties, actually roll up our sleeves, and actually get to work and develop that framework for the Human Exposome Project. Thank you.